Dear students, there is one more concept in your syllabus which is called as molecular docking. What is molecular docking? So there is a branch of bioinformatics called as biomolecular interactions, which talks about how the macromolecules interact with each other and how they function. For instance, a simple interaction I can tell you is about your antigen antibody complexes. So we want to understand how this antigen and antibody interact with each other. Now, let us understand how do you make these interactions work in a bioinformatics environment. So let us understand what exactly is docking. The process which brings two structures together is what is called as docking. It's a research technique for predicting whether one molecule can bind to another. If it binds, how much efficiently does it bind? So depending on these molecules, docking is of three types: protein-protein docking, protein-DNA docking, protein ligand or protein chemical molecule docking, which is used mostly in drug designing. The word dock was derived from the shipyard docks, where the ships are bought in and they are stabilized for a particular repair or maintenance. Now, docking is a computational process which involves prediction of favorable conformation of ligand within the binding site of a drug target. So, how do you understand this? If you want to understand this, let us understand what exactly is Emil Fischer's molecular uh, hypothesis of enzyme lock and key hypothesis. Like Emil Fischer has told that an enzyme substrate can fit like a key inside the lock of a door. The same way, the substrate will fit like a key in the enzyme active site. That's what is the Emil Fischer's hypothesis says. The similar principle is used in this case, where chemists try to accurately predict how a chemical molecule can sit inside a three-dimensional crevice of a target protein structure. So there are different components of docking process. One is you're trying to have a target protein. The second one is a ligand, if it is a protein ligand docking. And you're trying to use the combination of these two components through different types of algorithms, through different types of mathematical scoring functions, which I don't want to confuse you out, but want to give you a preview that how these molecules can be posed inside the active site so that they can stably sit. So if you observe the lock and key principle, in this case, the docking is called as rigid docking. In case of rigid body docking, the geometrical complementarity or the shape complementarity is considered inside the molecules. So what complex between a protein and a potential lead or a chemical molecule looks, and how strong is the affinity of these chemical molecules with respect to other candidates? Now, when you understand a molecular binding to a particular target protein, it involves a lot of thermodynamics association and thermodynamics function. As we have seen in case of protein modeling also, that the minimal energy indicates the stable configuration of the protein. Here, the binding energy is calculated in such a way that it should be minimal. So, the least the binding energy is more stable the interaction. So there are two types of docking. One is called as rigid body docking. The second one is called as flexible body docking. In case of rigid body docking, the receptor is held rigid and the ligand is also held rigid to explain the different degrees of translation and rotational freedom. So where at a particular point, the molecule will fit into the target protein. The second type of docking is called as flexible docking, a more common approach, which depends on the induced fit hypothesis of the enzymes. And here what we are going to do is we are trying to keep the rigid receptor, but we are going to make the ligand flexible. So in this case, when you do the docking, the ligand, due to the ligand flexibility, the ligand can attain different conformations and it can properly fit inside the target structure. So this is how you will understand the docking concept. And finally, this can be used to identify the druggable sites, the biochemical function of the molecules, the ligands, and other things can be obtained. Now, this animation will better show you what is the difference between the flexible ligand docking versus the rigid protein docking. Flexible protein docking versus the rigid protein docking. You can see the rigid one first. Here you can see the target molecule binding rigidly into the different crevices of the protein active site. But in case of the flexible, it is not searching different conformations, but it's trying to bend itself and try to fit inside the three-dimensional structure of the molecules. This is what is the difference between the rigid body docking and the flexible protein docking. So I hope these videos will help you for your examination. Thank you all.